All right. Well, good morning, everybody. So nice to welcome everybody to the Training Wheels Off support group. We're going to have our monthly guest speaker this week, Dr. Micheal Kaplitt. He specializes in neurosurgical treatment of movement disorders like Parkinson's and other things, and he uses deep brain stimulation. He's an expert in the treatment of various surgeries, general neurosurgeries. He uses high intensity focused ultrasound for the treatment of essential tremors, which was approved by the FDA. He is internationally recognized as an expert in gene therapy and molecular neurobiology. Well, that's a mouthful. And he is a leading figure in the development of new technologies for improving or restoring brain function. So, Dr. Kaplitt, thank you for joining us. He's going to give us a presentation, and at the very end, we'll have a open Q&A for those that want to ask him any questions. So, with that, I'll turn it over to you, Dr. Kaplitt.
Thank you very much. It's a real pleasure to join all of you. Thank you for inviting me. I know we have, I think, about a half hour is what it was on my schedule, and I have a meeting after this, so I would like to get right into it. I just want to say thank you for having me, and also thank the Ungers in particular for inviting me and for being so thoughtful and brave in trying to help move this field forward. And so what I'm going to do today is I'm going to tell you a few general points about the concepts of gene therapy. And then I'm going to tell you, because we have limited time and this isn't really sort of, there's really no ability to go into every possible different type of gene therapy that's going on. I'm going to really focus on this study that the Ungers are participating in. Just as an example, because it was the first and so far the most advanced of the gene therapy programs that have been done in Parkinson's, although there have been a bunch of things that have been done over the years and continue to be done in Parkinson's. So let me share my screen here. Okay. Full screen. Everybody can see this? Yes?
Yep. Yeah, we can see it. Thank you.
Great, thanks. So as you heard earlier, I'm Mike Kaplitt. I am a functional neurosurgeon here at Weill Cornell in Manhattan, in New York. I have been in practice for 23 years. I have both an MD from Cornell and a PhD from Rockefeller University next door. And I started my interest in gene therapy when I was a PhD student back in the late 1980s, and did sort of the earliest work in gene delivery in the brain. And one of the things that I became fairly well known for was having developed as a graduate, at the end of my graduate work, a technology called adeno-associated virus, or AAV, which has now become the most popular approach to doing gene transfer in the brain. So gene therapy fundamentally is a concept where we use modified viruses to put genes into certain populations of cells in the brain to try to improve those cells in various ways. So in this particular case, is to try to improve the function of cells and improve the circuitry that they control. In other cases, you're trying to correct a genetic defect if there's a genetic abnormality, and we've done this for a few genetic diseases that we've worked on in the brain. And in other cases, you're trying to use genes that might protect cells from dying. And then in some cases, you're trying to do all of the above. So the idea is to use these modified viruses to give cells essentially new instructions. As most of you may remember back from your, school days, genes are the instruction sets for the cells. They tell the cell, they're like the map that tell the cells how to function. And so by giving a cell a new gene or a different gene, you confer upon that cell the ability to start producing a particular protein that gene encodes, that gene gives instructions for, and that will change the function of the cell, hopefully for the better. That's why it's called gene therapy. That's the idea. We use modified viruses. So I think I have a slide here, right? So we use modified viruses because viruses are nature's gene delivery vehicles, right? So whenever we get sick from a virus, it's because of our immune reaction to the virus that we're getting sick. That's why if you get really sick, sometimes you'll take, anti-inflammatories or even a steroid pack, maybe a Medrol pack or something when people have really bad viral problems. And that's because you're trying to reduce the body's reaction to the virus. But the virus itself, in most cases, is a very simple thing. It's, it's actually a philosophical debate as to whether viruses are alive. Bacteria are alive. Viruses are not capable of reproducing themselves on their own. They don't have the machinery necessary to survive and reproduce on their own. So it depends on one's philosophical definition of life. Bacteria, when you have a bacterial infection, bacteria are actual cells that can reproduce themselves. But viruses need to get into your cells and make more of themselves. So the way they cause disease is they get into your cells. They then co-opt the function of your cells, and then they produce more viruses that then get released, and then those can infect other cells and make more viruses and then get released, and that causes havoc, usually because of the viruses, or usually because of the body's reaction to the virus. But on the other hand, over millions or billions of years, nature has really done a lot of the work for us in terms of optimizing gene delivery, because these viruses have been selected over these millions of years to be incredibly efficient at putting their own viral genes into your cells. That's what they do. They get inside your cells and put their DNA inside your cells. And depends on what the cell is. Different viruses are very good at getting into different cells. So with gene therapy, what we, those of us who sort of helped found the field realized, decades ago, was that if we could take the viral genes out and just put a therapeutic gene, a gene that might make a disease better, whatever disease that may be, if we can put a so-called therapeutic gene inside that viral package, now that viral package is able to deliver that therapeutic gene into the cell, and there's no viral genes in there, so it doesn't make more of itself, it doesn't spread, it doesn't cause disease. There's still the slight possibility that the particles that are delivering them, because they still have some viral proteins on them, could provoke a bit of an immune reaction. But generally, it does not do much if it doesn't reproduce itself. And that's what we found over the years. So that was the major advance back when I was a PhD student in the late 80s, was the idea that we could do this. Our big advance was that in those days, people were using viruses that were known to cause disease in that area. So for example, there are viruses that cause lung infections, and people interested in cystic fibrosis, which is a lung disease, were using viruses as gene therapy agents in experiments to deliver genes to the lung. People who were interested in diseases in the brain were using herpes viruses, which naturally infect the brain. That's why, when you get a cold sore, that's a form of a herpes virus that just sits in your, or when you get shingles or whatever, which is another variant, chickenpox is a variant of the herpes virus. They will sit there in your nerve cells, in your neurons, and then reactivate. And that's why you get these rashes, because they go from the nerve, out the ending of the nerve and infect the local skin and cause inflammation when they reactivate. So people were using, and that's what I did my PhD on, was using a form of herpes virus in the brain. But then we had this revelation that if we're just trying to deliver the gene into the cell and we don't care about what we call a productive infection, spread and making more viruses, etc., then in theory, we might be able to use any package that's capable of getting into the cell. It does not have to be one that normally causes disease in that organ. And that's where we came upon this particular virus called AAV. So adeno-associated virus is a very small virus that has never been associated with causing human disease. It is in a class of viruses that have relatives that can cause some pediatric blood disorders, but it's never been known to cause human disease. And yet we've all been exposed to it. About 85% of people have had some exposure to AAV, when you were children, because it, as the name implies, it hangs around with other viruses. I don't know what happened to my screen here. I apologize. I don't know what's going on with those marks. It's very strange. So, so that was our big contribution scientifically initially, was to show that a virus that is not normally known to get into the brain can actually get very nicely into neurons very efficiently and can be very safe and can last. The other viruses that we were playing with in those days only lasted for a few weeks or maybe at most a month or two, and then they went away. Whereas this virus happened to have a property where it could stay pretty much indefinitely, and we proved that. And so that was the big advance. So then we showed in this paper many years ago that this could actually, that was the paper where we showed that this could actually work in an animal's brain and can be effective. And that became literally the foundational event in this field of gene therapy in the nervous system, because now pretty much every single human study that's been done or is being planned uses some variation of this AAV vector. And every single neuroscience lab around the world is using it to manipulate genes in animal brains to try to understand how they function. So then we came up with this idea, and there's not going to be an exam at the end, I promise. So I'm going to show you a lot of data because I want you guys to actually see some of the things that we can actually achieve in terms of human experimentation, the level of information we can gain from patients in clinical trials to help guide what we should do next. This is a very busy slide. On the left is the normal functioning. I don't know why. Is this on? Oh, yeah, I don't know. I guess I just got rid of it.
cleared it for you. It's all set.
Oh, thank you. Oh, it was on your side. Okay, yeah, I didn't know where it was coming from. Sorry, I apologize. So, on the left side is the circuitry that is responsible for controlling movement. And DA is dopamine. And I'm sure you're all familiar with dopamine and the problems of the cells that are lost in Parkinson's. So this is kind of the way the circuitry functions normally. So you have dopamine and it affects the area that it goes into, the striatum, and that ultimately regulates all of these other structures. One of the key ones that we do surgery in, so I in my day job, I do deep brain stimulation and focused ultrasound treatments for patients with Parkinson's disease and tremor, and have been doing that, like I said, for 23 years for DBS and 8 years for ultrasound. But for the gene therapy, it depends on what the application is. But in this case, we're going into the subthalamic nucleus. So what you can see on the right is that when you lose these dopamine cells where it says DA, so now they're no longer doing anything, you get a dysregulation. So all these black lines some become bigger, some become smaller, and that's because you get a dysregulation of this whole circuit. It's just not functioning normally. And so when you take the drugs that you take that make dopamine or that increase dopamine in the brain, they will try to restore the function of the circuitry by going to the area that it's needed. But over time, the disease progresses, people develop resistance to the therapy. They need more and more dopamine. I'm sure some of you are familiar with this. You need more dopamine. You're not responding as well. You have side effects because of the high doses that people need to take, and the brain was never really intended to take drugs at that level because it's usually released in very small, regular packets, not these big peaks and valleys that you get when you take it by mouth. And so you get all these problems over time with this circuit becoming dysregulated again after doing pretty well on medicines early on. And so the question is how to fix that. So, one approach to gene therapy is to try to put the genes into the target of the dopamine area that make dopamine and let the brain make more dopamine again. But for most patients at this stage, making more dopamine is not the problem. The problem is the response to dopamine. So our approach was to say, look, we know that we do surgery in the subthalamic nucleus, which is the STN here, which is the area that you go in, that we do deep brain stimulation in. So we know how to do that surgery. And that's a big problem in a lot of these experimental therapies, is not just showing in animals that you can get a good result, but actually how do you do this in patients? Because there are a lot of things that we can do in animals that we can't do very well in patients. Well, we know as surgeons how to do subthalamic nucleus surgery. It's very easy. We also know that, as you can see where this box at the bottom that says STN is, it's well away from where the dopamine problem is as the disease progresses. So if we could try to use that as a means to try to restore or even restore the normal circuitry or even create better, more robust circuits, that maybe we could do something useful for patients. There's also some evidence that we and others have generated that you might actually somewhat slow the disease progression. That's not the primary purpose of the studies that are going on now, but there is some evidence that you could actually slow down the degeneration of the dopamine neurons that are left by doing the particular gene therapy approach that we're doing. So that was our idea, was that if we put a gene in and that's where the name AAV-GAD comes from. GAD is called glutamic acid decarboxylase. It's an enzyme that makes, that allows you to make GABA, which is the major chemical in the brain, the major neurotransmitter that slows or stops neurons from being hyperexcitable, right? And as you can see here, there's GABA everywhere, right? There's all these connections that are primarily responsible for releasing GABA. And so if we could restore this normal balance in the circuitry, then we could maybe normalize the circuitry without the need for these dopamine cells. So we did originally a so-called phase 1 study many years ago. And I did that in 12 patients where we only treated one side of the brain because it was the first time gene therapy had ever been done in the adult human brain for anything. not just Parkinson's, for anything. So the FDA that regulates this was obviously very nervous that something bad might happen. So they said you can only do one side of the brain. So we only treated one side in those patients, and they did well. And the side that was treated seemed to get better. The other side obviously didn't do so well. So as a group, they did pretty well. So that led to a second study that was a so-called randomized blinded study where some patients got sham surgery, some patients got the real treatment. And there we did it on both sides in the second study, and that was done at multiple centers. And so that was published, and we saw, as is common, and I'm going to show you something interesting about that in a minute, in almost every surgical study of Parkinson's disease, there's been some element of a small, what I call sham effect. It's not even necessarily a placebo effect. It doesn't mean the patient themselves thinks they're better. There are a lot of reasons why patients may score better, even if they didn't receive the full treatment. So you see that patients did do a bit better with the sham, but they did considerably, much better when they received the actual therapy. A negative score means that those scores that they do on you when they test all your different movements, your tremors, your hand tapping, your foot tapping, your walking, all that stuff, that composite score was better. As the number gets lower, it means you're less disabled. And so that was significantly better over time. This was the first gene, and to date, the only gene therapy ever that has shown a positive signal compared to a sham group in the same study. Other things that have been approached, like these dopamine therapies, et cetera, looked promising in early phase studies, but didn't bear out in later phase studies, even though there is still interest in pursuing some of these things, and many of them are ongoing. So then, and also we did a so-called responder analysis, meaning how many patients did particularly well. So we set a 9-point improvement as being a so-called responder. It doesn't mean you weren't better. If you were 7 points improved, you might be better, but we wanted people who were really improved. And what we saw was, again, a substantial difference between groups. At 6 months, 50%, at 12 months, 63% of patients exceeded that, sort of very good responsive standard, whereas in the shams, very few, was only 1 or 2 patients. So that suggested clinically that this thing was helping. So then, and then this is also dyskinesia, which some of you may suffer from. We showed that we could reduce the amount of time that people spent in dyskinesia. That's in the red here, compared to the shams, where they really didn't change. And by 12 months, they were a little bit worse, because obviously as time goes on, dyskinesias can get worse if you're not treated. This is the on time, the number of hours of the day that you spend in your good or bad state. That in a small study, this was a relatively small number of patients. There were give or take 20 patients per group, so it wasn't a huge study, even though it did meet all of these statistical criteria for a difference between groups. Here, there was a significant difference across the study in the so-called on time. At 6 months, there was something about these are based on patient diaries, which can be very variable. Those of you who participate in studies know that sometimes you're supposed to keep like a daily log for 2 weeks of what's going on. Some patients forget, and on their way into the visit, they'll kind of fill out what they remember. So these diaries can be a very difficult way of recording what's going on with patients in terms of how many hours they're spending in a good or bad state. But nonetheless, there was a significant improvement in the amount of on time. But interestingly, on and off time, meaning when your medicines are working and not working, was not an entry criteria for this study, the previous study. So this was despite the fact that we didn't actually enroll patients based on whether or not their on time was bad. And so if you looked on an individual basis in the treated patients, if you compare the amount of on time at baseline, which is on the x-axis, so how many hours of on time they had when they entered the study before they were treated, and what their change was, you can see that in those patients that were worse, that's where this cut line is in the middle. Those patients who had less than 8 hours a day of on time did particularly well and had an average of about 4 hours of increased on time. Whereas the patients who had a good amount of on time and didn't have much off time, right? I mean, you can see here, there are patients that had 13 or even 15 hours of on time. You'd say, well, if they're on for 15 hours a day and we only stay awake 16 or 17 hours a day and then you have to sleep, then why did they even get into the study? Well, obviously they had other disabilities that met criteria for that study, but those patients could not get any better than that. So we did feel that if on time was a big problem, that this in theory might help with that. So that was that study. This was complications. Again, there was an improvement in complications, a reduction in complications. You see there's a % improvement in complications in that group. This showed that only the sham group had like kind of worsening of Parkinson's disease as a complication. This is while everybody was blinded, so they didn't know. Now, here's the interesting part. So in addition, and the Youngers know about this because they've been doing this, we also did brain imaging, and we did what's called PET scans, which looks at the uptake of sugar in the brain and looks at, and so we can actually see these networks, these brain networks that I showed you earlier, and see how they're changing. And so my colleague, who's an outstanding expert in this field, David Eidelberg, who's at out in Long Island at Northwell Hospital, he's one of the leading experts in doing this. He analyzed it. He used an algorithm, a way of analyzing these scans, where he could actually look at the networks that were created specifically by the treatment. So this, as you see here, these are, you see multiple colors here, like blue and red, etc. These are different areas of the brain changing. So blue means they're less active, red means they're more active. And trust me when I say that the blue and the red are going in the right directions here, right? The areas that are blue are areas that were too active before and we wanted to quiet them down. The areas that were red were less active before and we wanted to increase them. That's that, you know, based on the areas that we need. So that's what he could look at and say, were we creating new brain networks with this treatment that improved function? And the answer was yes. You see in the red a dramatic increase in these, you know, better brain networks, and you don't see much in the sham patients. And these are actually the individual patients, and you rarely see this in this graph where there's almost no overlap, even on an individual patient level, that most individuals that were treated had better brain networks and most of the shams did not. And it correlated well with their degree of improvement. That's what this graphs at the bottom show, is that the more negative scores, meaning the better improvements, patients who had 15, 20-point improvements, had the most change in, in this better network. I'm going to skip. Oh, no, this is important, actually explains this. So we asked a really interesting question that didn't occur to us at the time. When we did our original study where I only treated one side of the brain, we also did these PET scans, but we knew nothing about these networks that we found in this study. So now that we understood it, we went back and reanalyzed the original data from years earlier because we had it. And what we found was that in the original study, we did 3 doses, a low dose, a medium dose, and a high dose. The high dose was the dose that we took forward into the last study, and that's the same dose that we started the current study with that the Youngers participated in. When we went back and looked, if you look on the left, you see that this network was really only present in the high dose group. The medium and low dose patients didn't have much at all. And so that said to us that the dose that we called the high dose from the original study may have been just barely scratching the surface and may have just been barely getting into a therapeutic area. So based on this data from our human patients, we were able to conclude and convince the FDA that it would be worth testing an even higher dose. And that was one of the purposes of the study we're doing right now, where we are testing in this current study that the Youngers participated in, an even higher dose compared with this original dose that we did before, to see whether that is better or not. And we don't know the answer to that yet because that's ongoing. Now, the final thing that I want to show you is that we asked a similar question, and this to me is one of the most fascinating things we've ever done. So we all talk about the placebo effect, the sham effect, whatever in surgery. So we said, is there a brain network we can identify from these scans, from these PET scans that are looking at function? Is there a network we can identify that actually explains this placebo or sham effect? So my friend David asked a very interesting question. And these are all computers, right? So you're not picking and choosing. You're just asking the computer, you're asking mathematical questions saying, are there any brain regions that change in sham-treated patients that got better compared to those that didn't? And it turned out there was. And it was a whole bunch of areas that had nothing to do with movement. These areas that are lighting up in red are, this is an area that's very responsible. This it said subcal 25. That is a central area for depression. Same thing with this area of the anterior cingulate. So these are areas that are responsible for sort of mood, etc. And that's what lit up in patients who had this so-called sham effect, which means that it was much more of a psychological effect than it was a movement network effect. And so if you look here, this is a busy slide, but this is the most interesting thing I think we've ever done. If you look here on the left, at 6 months, when the patients did not know what they got, what you can see is that the sham subjects in dark gray showed a lot of this brain network that was responsible for the sham, whereas the treated subjects in light gray had none of it because they were the ones who had changes in the actual movement circuitry. Then at 12 months, when we told them what they got, so now everybody knew, look what happened. The sham subjects showed less of this brain network. It went down in the dark gray group. I interpret that as meaning that they knew that they found out that they were sham, and they were kind of disappointed that they received the sham therapy, so they're not having as much of this anymore. Whereas the treated subjects in the light gray started showing some of this network because they were told that they got the treatment and presumably were pleased about that, right? So it was really fascinating because you're seeing how these networks change and explain what's going on biologically. And we know that a placebo effect is a component of every treatment we do in medicine. There's nothing wrong with having it. You just don't want your entire treatment to be due to a placebo effect. But having a real effect and a placebo effect together is part of what we do every day in medicine. Everything we do has some element of that. It just shouldn't be the whole thing. So there's nothing wrong with it. It was just fascinating. I think I'll stop there because that kind of explains the background of what we've done here. Now, there are also gene therapies. I've been doing that myself to try to correct genetic forms of Parkinson's disease, particularly the so-called glucocerebrosidase form. We're not doing that anymore. We did some patients in that study. That's ongoing. There are gene therapy studies that people are doing to study how to stop the dopamine neurons from dying. And we're doing some of that in my laboratory, but not in patients right now. So there are a whole host of ways you can put genes in. But this is the one that we have the most human data on, and that explains the study that the Ungers participated in and why we did it and where we're at with it. And so I think I'll just stop there and answer any questions that anybody may have.
You could unshare your screen, Dr. Kaplitt. That'd be great. Thanks. All right. So anybody have any questions? Just raise your hand, and I'll go ahead, Greg Unger. I unmuted you. Go ahead. Go ahead, Greg.
Is there any particular order that the vector has in treating the symptoms? For example, last to appear and then first to get treated?
Say that again?
Is there any particular order that the vector has in treating the symptoms? For example, the last symptoms to appear are the first ones to get treated?
That's a good question. No, not necessarily. I mean, we do have some evidence that certain symptoms may be more responsive than other symptoms. So it is correct that it is possible that when I say you, I mean one, because I don't want to talk specifically about you. But so when I say you, assume I mean just anybody.
Sure,
it is entirely possible that someone treated might notice a real improvement in some symptoms, less improvement in others. And it may be a coincidence that correlates with the order in which one develops symptoms. So it is absolutely true that certain symptoms might respond more than others, but it's not based on when they came about. It's what those symptoms are, because these networks tend to influence certain symptoms differently than others.
Okay.
And we're still trying to understand that better, but we have some evidence from the past study that that's true.
Sure.
And that's true of pretty much and that's true of everything we do in Parkinson's, whether it's deep brain stimulation or anything else. We always tell patients, well, this might get better, this might not. These are the discussions we always have. And that comes from studies like this so that we can understand how to position it best for particular patients, right? And that's where we go. One of the things that I've felt very strongly about in my career is that, there are a lot of people in our field who want to try to have the one-size-fits-all cure for everything. And that would be wonderful. But there's very little of that in medicine, other than antibiotics to treat infections. And even there, some work for some infections and some work for other infections, right? But we don't cure a lot in medicine. We cure infections, we cure broken bones. That's about it. Everything else we treat well, right? Heart disease, we don't cure heart disease with a stent or bypass or drugs or whatever. We just make it better, but we don't cure it. So the idea that we're going to have one thing, I'm not implying that this is going to be the cure for Parkinson's. I don't think anything's going to be the one single cure for every patient. Part of what these studies are designed to do is, first and foremost, confirm that these things actually are biologically valid and doing something useful, so that we don't approve things, get FDA approval and treat people with things that have no biological validity. But then just as important, as your question implied, is to understand who are the best patients that might respond best to this therapy, because that's exactly what was done over the years, whether it's with medications, whether it's with deep brain stimulation, with other things that are going on like ultrasound. It's these studies that allow us, when we sit in a room with you and give you these speeches about, we think this will get better, but this may not get better, comes exactly from these studies to understand that.
Sue, you had a question?
Am I unmuted? Yes, Doctor, I was just wondering if any of the members here are interested in any future studies. How would they go about, qualifying or applying?
I should have mentioned this, so thank you so much for being more attentive than I was. So, because I going into this, I wanted to mention this. So, there are a variety of gene therapy studies going on in the world, and if you go to clinicaltrials.gov is a real repository, not just for gene therapy, for any trials that are going on of any kind. They can be device trials, they can be cell therapies, they can be gene therapies, they can be drugs. Any trial that's going on, you can just put in Parkinson's, for example, and clinicaltrials.gov will spit out a whole list of everything that's going, tell you which studies are enrolling, which are not. For this particular study that the Youngers are involved in, this initial study where we're testing the higher dose, mostly to see if it's safe, because no one ever tested that high of a dose before. So that was the purpose of this study, was to see if it's safe and maybe get a hint of whether it works, better or not. But it wasn't designed with enough patients to really prove that. If the end results of this are encouraging enough, then another study is planned to hopefully be initiated early next year, so not too far out, where we would do a very large study on a large number of patients to actually prove what we're, sort of testing here. And so I'm hoping that might start enrolling early next year, so not too far off, but I can't guarantee that. But as this goes on, I will probably have a better idea by October, November of what the plan is for that. So not too far off. So if anybody were interested, I certainly would be happy to talk then. But and I'm certainly happy to talk to anybody about anything, whether it's standard therapy or experimental therapies, whether I'm doing it or not. I'm more than happy if any one of you as an individual wanted to reach out to me. But certainly when it comes to this, I'd be happy to talk to anybody going forward. But that's a resource. Clinicaltrials.gov is a general resource that will show you everything that's going on.
Great reminder. Thank you so much, Dr. Kaplitt. Anybody else have a question for the doctors? Edie? Go ahead. I unmuted you, Edie.
Okay. I was just curious. You mentioned the adeno-associated virus. I was just wondering, where does that come from?
A great question. People have asked me that before, and the first time I was asked, I was kind of thrown. Sorry, I just want to let my other meeting know that I'm going to be a few minutes late.
Appreciate you taking the time, Dr. Kaplitt.
No, not at all. I want to finish this, so. Yeah, so the adeno-associated, it's a good question. And I can only partially answer that, right? So it is synthetic in what that means is we take cells in a dish and we take pieces of DNA that we can put into the cells, and we have ways of doing that chemically, just get the DNA into cells. In a dish, we have various chemicals that will allow the cells to take up fragments of DNA. So we put those fragments of DNA into cells, and once they're in the cells, they start producing things. And so some of those fragments of DNA will produce the AAV package that I talked about, the proteins that form the coat or the sphere or the whatever, the hexagon that I showed you earlier. So some fragments of that DNA will start producing that. And then another fragment of the DNA is the actual gene that we're interested in delivering. And that will, so that coat, or what we call a capsid, will actually naturally form around fragments of the gene that we want to deliver therapeutically. And so the cell will start acting like a little factory pouring this stuff out. So we synthesize the genes in a lab, in a machine that allows you to basically synthesize fragments of DNA. And then we put them into these cells in a dish that we've carried for a long time. And then they ultimately, over a few days, start spitting out trillions of these packaged viral particles. And then we have ways of purifying them away so that they're absolutely pure. There's no cell components to them. The FDA has 1 trillion tests, as you can imagine, before this can be released for injection into the human brain. So it's an incredibly expensive process, and it takes a lot of time. It takes about 6 months to pass all the tests that the FDA requires to make sure that there's not a hint of contaminant left over from cells or other things that could, cause inflammation or cancers or anything that might be a problem. And there never is, and no patient has ever had any issues like this in our studies. We've never had a patient have a serious issue with inflammation or other problems due to these things.
Did that answer your question, Edie?
Yeah, thank you.
Okay. I had a question, doctor. So the treatment, it's delivered intravenously, or is it
No, it's directly in the brain.
surgically? Oh,
Yeah,
surgical.
this is
Okay.
surgically. So the operation that we do is very analogous to the operation for deep brain stimulation, where we make a small hole in the skull. You're in a frame that's holding your head in place. We make a small hole in the skull, and then we map out the area to make sure we're in the right spot. But then instead of putting in a big deep brain stimulating electrode and a battery that goes in your body and all this other stuff, we just put this very thin catheter or infusion tube. It's literally like the size, just barely bigger than a human hair. So much smaller than a deep brain stimulating electrode. And we put that in there, and over a period of several hours, we infuse it into the brain. In the current study, we sedated patients during the infusion so that it wasn't uncomfortable, while you're lying there for a few hours. But in the next study, we actually won't need to do that, because in the current study, we were using the catheter system as one that we will eventually use in any patient commercially if this ever reached the point of getting approved. But the pump that was infusing it was an experimental pump. We have now created a pump that will be usable by everyone. And it's, the pump before was huge. Now it's about the size of my palm. And so we will be able to close the skin and let the infusion happen in the recovery room. So you can be talking with your family and eating during the 3 hours while this thing is infusing, and then it can just be pulled out at the bedside. You don't have to go back to the operating room. So in the next study, and eventually in the commercialization process, if it ever gets there, and I'm not implying anything that it will, but if it did, the plan is to actually just implant these things, which takes about maybe an hour or so, and then do everything in the recovery room so that, and you never have to go back to the operating room, and then everything gets removed and you don't have anything left in your body at all, except a little cap that covers up the hole that was made, what you want, because you don't want the skin to kind of divot in.
Oh, that's very interesting. Thank you for clarifying that. Edie, do you have another question?
Yeah, is this a one-time treatment, or is it something you have to do multiple times?
Yeah, it's a good question. Our hope is that it's a one-time treatment. Again, we've never retreated anybody. There is some concern that if you retreat with the exact same product, the immune system might have been primed the first time to react to it, so you might not get as good of an effect the second time. But there are ways to repackage it so it's slightly different but does the same thing if you did need to retreat it. We do know that in animals, it lasts, the gene keeps producing what it needs to produce indefinitely. So the hope is that the benefit, that the gene won't go away. But the question is, if the disease worsens over time and the brain continues to change, will the brain develop some resistance to the therapy, even if patients do well for a year or two or something? And that we don't know. We have some anecdotal evidence from isolated patients that they did well for many years, but we don't know that for a fact yet, and so that's part of the ongoing studies. That's why we have a so-called long-term follow-up study where we're following patients for years, as long as they're willing to continue to be followed.
Okay, thank you.
Thank you.
Very good
Anybody
question.
else have a question?
I do. I do.
Go ahead, Drew.
So for the sham patient, are they going through the whole process of being, let's
Right.
just call it, deceived that they're
Right.
getting the treatment, and they walk out of there with a wrap around the head and the whole thing?
Yeah, correct. So the way we're treating the sham subjects is, so the sham subjects do not get actual brain surgery. They're not put at risk in that way, because we can't do that. It's unethical to go in. There is every time you penetrate the brain, there's a very, very slight risk of something happening, like bleeding or something like that. You're not going to do that in a patient that has no possibility of getting improvement, right? So that we've never done. The sham surgeries are, we make a small hole, but we don't go all the way through the skull. We go about halfway through the skull. So from the patient standpoint, they have the same incision, they have the same cap covering the hole, and they have a partial hole, but we don't go through the skull, so there's no risk of injury to the brain of any kind, right? And that has been deemed ethical, and that's what we've done, and we've never had a problem with that. But yes, if they are sham, they, are being evaluated for 6 months, and then if they were, then they wouldn't have gotten the treatment. at the end of the 6 months of the so-called blind, when the patients are done with the period, in this case, it was 6 months, where we're assessing patients for whether or not they responded, then the patients who did receive sham have the option to go back and get the actual treatment, and they would know they got it through the same holes. So, the way we do it is we plan the operation so that the holes are centered over the correct location where we would plan to do the actual operation. We just don't actually do it, but we go in that same area. We did all the planning and everything. So you would use the same hole in the same area and just go in and give them the infusion if they want it. It's their option. They're not required, but they can get that, or they can go on and do whatever they want.
Great question, Drew. Thanks for asking that. Anybody else have a question? All we have. Oh, Linda, go ahead, Linda. I unmuted you.
Thanks. How many people do you anticipate in your study?
In the next one, in the next study?
Yeah.
Well, if this works, I think we're talking about maybe 100 patients in the next study. That would be around the whole country, not just at a couple of centers. But yeah, about 100 patients total.
I think
And
you need more.
Somewhere around 20 or 30 centers. Pardon me?
I think you should do a lot more.
It's not so easy. Surgical studies, it's very hard to do. It's not like a drug study where you can easily do 1,000 patients. Surgical studies are always smaller because there's just no way.
That makes sense.
Practically on that many
Thanks.
people. I apologize. I really do want. It's just I'm 15 minutes late for other meeting.
No,
but
no
if there's
problem.
any pressing questions, I could answer it for like one more minute. But if there's any pressing, I'm also open. You have my email, I believe, because we communicated. So I'm happy to communicate with anybody who has any more interest in this or anything else, whether it's DBS, ultrasound, any of the things we do, experimental, not. I'm happy to communicate with anybody where I can help them.
All right. Well, thank you very much, Dr. Kaplitt, for coming today. Thank you for your presentation. Thank you for answering our questions. And if anybody has further questions, I'll let them know, give you your email address so you can communicate with them.
Thank you so much for having me, and thanks to the Ungers for having me and for continuing to, participate in this whole deal, because I know it's a bit of a distance and a challenge, and we've worked through some of that as well, but I appreciate it. And I do have to say, honestly, the questions have been really amazing. And the reason I say that is I do a lot of these types of things, as you can imagine. And rarely have I had every single question be so on point. And the reason I say that is because if you think about the answers I gave to each of your questions, these were very detailed, specific answers because you were asking very perceptive questions, right? So I was giving you new detailed information that I didn't do in my presentation. Usually I get questions where people want me to clarify things because they weren't following, or I spoke too fast or whatever, or things that, they'll ask me about other diseases or whatever. I've honestly almost never done one of these where every single question was so on point, and I really appreciate that. It makes it a lot more fun to do this, so thank you.
Oh, thank you very much,
Thank you.
Dr. Kaplitt.
Okay, take care, everyone. Good luck.
All right.